So now let's get into some of the safety as well as some of the uses that we do use biotechnology for now. So in the grand scheme of things, we've actually been genetically modifying things many years. We've been doing that with foods, with animals, through a process called selective breeding. Selective breeding is a process where we basically choose which of the animals or which of the plants that we're going to breed together to create more desirable traits. Genetic engineering, though, is something we do now. This is where we're manipulating an organism's genetic material because we're either adding something, deleting something, or transplanting genes from one organism to another. Now, we use what we call recombinant DNA technology safely, efficiently, and quickly to produce what used to be a very long, resource-heavy, and risky project. And that's why we're forming genetically modified organisms, also known as GMOs. Now, there's a lot of controversy surrounding GMOs, some of which is not really an argument or not really a good stance against them, but there are also some reasonable concerns as well. But first and foremost, we do need to remember that no matter what, DNA is DNA. It has the same basic setup. We're not introducing anything that is completely foreign in terms of its structure, and that the only thing that distinguishes DNA from other sections of DNA are the sequences of the bases, and that's it. Now, when you are looking at arguments for or against GMOs, some of the arguments are valid and others are not. In terms of GMOs being inherently just dangerous and that they can kill you, no. But there are some legitimate concerns when it comes to biodiversity and protecting that. Being concerned of these genes getting into wild populations of plants and how that could change biodiversity. That is a legitimate concern. There is a legitimate concern when it comes to food allergies, in that if you introduce a gene from something like a shellfish and you put it into another food source and that a person eats that food source and they're allergic to shellfish, there's a potential or a concern for allergic reactions. That is legitimate. However, that GMOs are inherently dangerous for anyone and everyone, that's not really the argument we want to make. There are plenty of spaces where we do see the absolute need for these, like in places where there is famine or issues with proper nutrition. GMOs could potentially help with that process of introducing crops to areas that will be able to handle the problems with the environment that area might be experiencing, or problems with nutritional deficiencies like beta carotene, for example. There are many positives as well as some drawbacks, just like anything else. And so it's up to us to consider the research, to consider the needs of the populations, to consider the needs of the planet when it comes to determining whether or not GMOs are appropriate for that given situation. Not all GMOs will be necessarily the most appropriate, and in many cases, GMOs will be a saving grace. It's something that is a mixed bag that we just want to take case by case rather than seeing them as a holy grail, but also not condemning them as an absolute problem for humanity. So, like I said, are the rewards outweighing the risks? It's a complicated thing. You're not going to be able to necessarily apply the same logic to every single case. It's going to depend on the individual GMO. What are the risks? What are the benefits? Right? But let's get into some of the uses that we do use GMOs for. Now, it's Iowa, and so driving around any time in the summer and into the fall, you see corn. But the corn you see is not the corn that always was. Ancestral corn actually looked like this picture we see above here. And so it was through selective breeding over long, long periods of time that gave us the big, juicy kernels of corn on the cob that we know today. So corn as we know it did not exist without human intervention. Corn as we know it exists because over time and over the generations, farmers selectively chose which plants they would breed based on the juicy kernels. Plants that had bigger kernels were selected for and bred. Plants that did not were not bred and were left out. And so over time, you see a push towards bigger, juicier kernels. Another example is golden rice, and this is where I said the benefits and the risks have to be weighed together. Golden rice is rice that forms beta-carotene. Beta-carotene is a nutrient that in some places there are big deficiencies and it can lead to problems with vision in those people. And so golden rice was developed as a way to potentially kind of fight against those health problems for people. And what they did is they introduced a few different genes from both daffodils and bacteria to allow the rice to build beta-carotene. And so it's called golden rice because the beta-carotene gives it a golden yellow color. Now, if we're looking at GMO crops in the US, most of the crops that we know today and that we consume today are GMOs. 86% of corn, 93% of cotton, and 93% of soybeans are all genetically modified, with the rest of those small portions being not genetically modified. Speaking of corn, we can look at BT corn. BT corn is a GMO where we took the bacterium Bacillus thuringiensis and we introduced this gene to produce spores from this BT bacteria that will kill the insect pests but doesn't hurt us. So it's something that is completely neutral to humans but will kill the insects that chew on the leaves. So if we look at caterpillars destroying corn plants, right, that's going to hurt the crops. But if we genetically modify that corn to introduce what we call BT crystals, which are poisonous for caterpillars, we put it right into the corn plant DNA. The corn grows as normal, and we don't have to use pesticides because then the caterpillars, anytime they try to eat the plant, they're going to die. There's also GMOs when it comes to fish. So when we look at farmed fish, larger salmon that have this growth hormone gene that keeps them going all year round end up very, very large, and we can harvest them faster than we can the what we'd call the wild type salmon. And so for something like this, there would be the concern that that gene could potentially get into the wild population if it somehow escaped a farm. But at the same time, having larger fish that grow faster, we can feed more people. So again, rewards versus risks. So what are some things that we do medically when it comes to GMOs? Really, the first commercially available genetically modified product was Humulin. Humulin was human insulin produced by bacteria. Now, previously, if you were diabetic and you were reliant on insulin, you had to get it from either cattle or pigs that were killed for meat. So in the process of the pork or the beef industry, they would extract insulin from them. However, this was expensive. It was not easy to do. And because the insulin was coming from non-human organisms, some people had immune reactions. But now, what we do during the chop, amplify, insert, and grow process, we took the human insulin gene, we inserted it into bacteria, and now the bacteria can make insulin that is literally indistinguishable from humans. It looks and acts exactly like human insulin and does not provide any sort of immune problems for the people taking it. And the Human Genome Project, if you look for it, genome.gov, this was another project that we did where we sequenced the entire human genetic material, the entire, what we call human genome, so we could learn more about our genes. Now, back in the days, we thought that this would be an end-all, be-all, and we would solve cancer, and we would solve all these different diseases. However, we now know that it's much more complicated than that, but we still learned a lot. And through that process, we developed technology to allow us to sequence genomes even faster and cheaper. The original Human Genome Project was extremely expensive, in the millions, if not billions of dollars. Extremely expensive. However, now we can sequence a genome for about $1,000. So we are making leaps and bounds of progress when it comes to understanding the human genome and genes in general. Biotechnology does help us with things like identifying and diagnosing genetic diseases. It's another aspect of biotechnology in human health. And through that process, we can use what we call genetic counseling, where if you and your partner are concerned about risks of certain genetic disorders, you can go through a process called genetic counseling to determine the risks of whether or not your kids could have those disorders. So looking at, could a child have this particular disease because of their parents' genetics? Could the child be born with a genetic disease? Could they develop one later in life? So these are things that would be we would go into in genetic counseling. Now, there are ethical questions about this. Do you think that this kind of consultation is actually helpful? Do you think it should be encouraged? If it's a risk and someone thinks, okay, there is a risk I'm not going to have children, that's a huge behavioral and lifestyle change that this person's making for something that may or may not happen. However, for some of these genetic disorders, the care is very, very heavy. It's very hard to care for some of these genetic disorders. It's very heartbreaking to care for them, and it can be not just emotionally draining, but financially draining as well. And so it's a big question of whether or not should we know all of this information, or is this something that we just have to find out as we go through the process we call life? Now, clones of cells, tissues, organisms, and whole organisms now can be developed. But of course, there are ethical challenges in addition to biological ones. This is because we need to determine where we draw the line in the sand of what is considered ethical and what is considered unethical. Do we allow for these processes to happen on any organisms? Do we allow them to happen on anything except for humans? Do we put certain stipulations on it where you can only use it for these certain reasons, or maybe other reasons? The questions are vast, and it's questions that we're answering every day. But another thing that we can do is that we can use it to identify people. Now, this is especially used when it comes to crime and forensics. Because DNA is totally unique to us, except when you have an identical twin, DNA does get used to identify criminals. Now, having your DNA on hand and looking at it does not identify you as you. There needs to be a comparison. But if they compare DNA samples that were taken from like a location, like a crime scene, to samples taken directly from that person and they match, we can say for a fact this person was at this crime scene within a reasonable period of time. Why is it unique to us? Why can we use DNA in that manner? Remember the sources of genetic variation. Crossing over that happens in meiosis 1 in prophase 1, the random assortment of homologs during metaphase 1, and the random selection of eggs and sperm to be fertilized. These three things together make it, we'll call functionally infinite different combinations of DNA.